I just read it, but. I think it's video 10. No, I already seen it like 4 times. What video do you want to see? How do you want to get into that? Where is it? I found it. She said like you're not even actually
Like, did she just send that?
Yeah, like maybe 10.
No, I'm trying to grab something to my bag. I feel like they're all pens.
Oh, okay.
Oh, honestly, I feel like I wasn't that tall. Look at it. Where are you? Right there. Where did you get your light pencil? Right there. Can you put it in slow motion? What is slower? 0.5. Zoom in. Look at my reaction even after. Like, it wasn't even thinking to have it on that spot. No. Okay, but you gotta think about it. We also did a Where are you? Right here? Yeah. That's what Andrew said. But I feel like people don't always look like this. Even like really like give like a whole direction. It's so skating. It's so skating. Then I'm like, looking at her like she's just looking at my leg. Who else is looking at your leg? Heavy. Okay, I kind of like this transition and like a ignore my face, but it is fun. I think it's fun. So,
Okay, but she's on something that she thinks you're gonna press that thing happens without
you can't. You're literally.
I tried telling her.
She said, well, you're gonna catch it perfect. No, you're not.
No, we're not. And
You expect Sophia? Yeah, we can't even do it.
then we should literally do it out of the thing before, right?
And it would be easier because you can twist down and then just drop your foot and the other one's already in.
Your face is not.
Oh, just I had to look at it for so long. I get it. Oh, no, actually, what did I learn? What? If you like get like your and then I go like I usually have like that because you're like okay. Are you ready? I'm gonna video and we have I just don't know if I had to look up my eyelash because it's working in myself. It was like I think if you have a general. Like I'm still going. I'm just I just walked yesterday. I'm already grabbed him. Look, I've already grabbed it. I already have the foot right here. Yeah, we have to wait longer. Yeah, I know. Like, why am I grabbing the foot right here? I don't know. I
guess yesterday.
Also, why is Megan still holding this foot right here when I'm grabbing it?
I mean, yeah, for you.
I think it's
I'm kicking you. She's telling me it's not me. Yes, it is because she's still holding this foot and I've already got the second foot. Okay, we're gonna show Heavy that. See? Look, she's just not letting go. Wait, actually, that's Lauren.
maybe I'm kicking more.
This is one stance.
Oh, is it? We just went to the work to get fish.
Isn't this your hand right here?
Yeah. Maybe I'm kicking.
That's getting stuck.
Guys, and that, I think that was just, we never learned anything bad. Like, it doesn't happen like that.
Why am I holding on to this which I'm already here? Maddie's still holding the other plane. Lauren has this one too, so.
All right, guys, good morning. Wakey, wakey. All right, I'm gonna take attendance. Brianna Aiken. Can I just get a hear from you guys? Because if my head hurts and every time I look up and down, it's like causing my migraine to get worse. So I just want to like hear you and then I'll check you off. Drew.
Here.
Bria.
Here.
Henry.
Here.
Anna.
Here.
Or Anna, sorry.
No, you're good.
Nick.
Here.
Abby.
Here.
Hannah.
Here.
Rosa.
Here.
Matt.
Here.
Jillian. Mia.
Here.
Annie. Brianna H.
Here.
Brandon.
Here.
Sadie.
Here.
Jessica.
Here.
William.
Here.
Marissa.
Here.
Anna. Sophia.
Here.
Aubrey.
Here.
Matthias.
Here.
Colin.
Here.
Olivia.
Here.
Braden.
Here.
Amber.
Here.
Katie.
Here.
Kennedy.
Here.
Ben.
Here.
Ava.
Here.
Kendall.
Here.
Emily.
Here.
Lauren.
Here.
Jen.
Here.
Isabel.
Here.
Corwin. Okay, cool. I'm very impressed. This class is a lot bigger than it was last year when I taught it, so. Okay, so just a reminder, we're gonna finish up chapter 4 today, and your next worksheet is due on Friday. It's gonna be on chapters 3 and 4. So if you come to class, you participate, you'll get the 10 points. If you are not going to be here, you have to email it to me by Friday so that I can give you the points. All right, let's go through to slide 23 where we left off. Okay, so we talked a little bit about, doing how to grow bacteria, either on, a spread plate or doing a dilution streak. And so remember, these are individual colonies. And so this is the goal, you want to see these individual colonies. Just to kind of, yesterday in lab we talked about, the pepper infusion and wanting to eventually isolate a pure culture. Does this look like a pure culture? How would you tell if it's a pure culture? So what we look for is how many different colony types we have. Are these colonies all the same type of bacteria, or are they different? Yeah, they're all the same. They look the same. You look for color, you look for, how raised or bumpy they are, you look for the edges. So these all look like the same type of colony. It all came from this bacteria. We're just spreading it and diluting it. So this is a pure culture. So in your pepper, if you just get one type of colony, you're looking for how many types of colonies you have to see if it's just one pure culture or if it's a mixed culture. Does that make sense? Okay. So when you do your peppers, if you see different colors of colonies, different kinds of colonies, different, some are bigger, some are smaller, then you know that there is going to be more than one bacteria growing in that liquid culture. Size is also a big one for colonies. So these all are the same size. Make sense? Okay, beautiful. So there's different types of media that are used to grow bacteria. Complex media is this really nutrient-rich media. It has everything you need in it. It has amino acids, peptides, vitamins, sugars, nucleotides, but you don't know the exact amounts of everything. So it's poorly defined, but it provides many of the building blocks for the majority of cells to grow. It's a complex media. So it's nutrient-rich, but poorly defined. The next one is minimal media, kind of the contrary of that, of the complex media, where it only contains nutrients that are essential for growth for a specific microbe. So the bacteria will grow very slowly because we're forcing the organism to have to synthesize its own essential components. We're only giving the nutrients that are absolutely essential for growth, for life. But through the cell's own metabolism, it has to make the other things. So we're kind of just focusing on allowing just that one type of microbe to grow. So you give it the bare minimum to survive, and then it forces its metabolism. Remember, their genes are very plastic. They turn on and off genes all the time. So we put them in an environment where we give them just the minimal amount of nutrients to just survive. Then they turn on these genes to then make the other things in order to grow. Does that make sense? Enriched media is essentially complex media where you enrich it with specific components. You add some specific things to it. So, you know, you have complex media, can grow a lot of different things, but if you want to make sure that you're growing something in there, then you can add a specific component to it to make sure you get that specific growth. Okay. Selective media basically favors the growth of one organism over another. So selective media essentially it makes sure that one organism grows and it kills off another. So one of the things that you can think about when you're thinking of selective media, you know, we talked about gram-positive and gram-negative cells, and so selective media will, you know, we can have selective media for just gram-negative cells or just gram-positive cells, so it favors the growth of one, it doesn't allow the other to grow. Differential media, instead of killing off one bacteria, both of them can grow, but you're exploiting differences between them. So they both can grow, but they grow maybe differently in terms of one grows as a red colony, one grows as a golden colony. So you exploit those differences between the two, and so the differential media allows you to differentiate between them. They both can grow. So it could be a color change or something if one organism can use a certain carbon source or whatever it is. So they grow equally well, but it exploits those differences. Questions on that? Okay. One of the media that we use a lot in clinical microbiology, and I kind of actually brought this up a little bit yesterday when I talked about E. coli being in water for fecal contamination. So this is you know, a similar type of plate that you can grow things on. So this is called a McConkey plate. You don't really need to remember that right now, but basically only gram-negative bacteria can grow on these McConkey plates, and lactose is the carbon source in there, so it's a selective media, because only gram-negative can grow, but not gram-positive. However, within the gram-negative species, there are different species of gram-negative bacteria, so it's also a differential media because it allows more than one gram-negative bacteria to grow, but they grow as different colors because some of those lactose fermenting gram-negative bacteria are able to use that lactose and they turn red, and then the non-lactose fermenting bacteria grow, but they grow as, they're not able to break down lactose, so they grow as just regular like yellow colonies or uncolored. So does that make sense? So some media can be both selective and differential. It's selecting for gram-negatives, but within the gram-negatives, some are lactose fermenters, so they are able to break down that carbon source, and it shows up in the color that it grows in, and then it can grow gram-negative non-lactose fermenters. Questions? Does that make sense to you guys? So fun fact, these red ones are E. coli, and this other one is S. enterica. So this is one of the ways that if you were to take a water sample and you want to know if you have fecal contamination in there, you can, and remember E. coli is gram-negative, and so you put it on a plate, and if you see this red color, then you know it's fermenting lactose and you know there's E. coli in the water. Okay, so, you know, I pretty much have brought this up in every lecture so far, but there's some microbes that are unculturable. Some species are adapted so well to their natural habitat that we don't know how to grow them in the lab. And so, you know, some of these unculturable organisms, and that's quote unquote, because obviously, you know, science is ever changing and there may be a day we can culture them, but some of these organisms depend on factors that are provided by species that also cohabit their niche. Some are social, they, you know, refuse to grow unless they're with their buddies. So microbes in the environment are probably providing something to that unculturable microbe that we haven't figured out yet. Okay, so there are a lot of different ways to count bacteria. Bacterial counting is pretty important, and so we'll definitely do some of this stuff in lab. But there are many reasons why it's important to know the number of organisms in a sample. Counting the organisms, you know, is hard because they're invisible to the naked eye. So there's techniques that measure a different physical or biochemical property that allows you to count the amount of growth. So, you know, there's cell density is basically how many cells are in the media. You know, you might use two different techniques and it might not be exactly equal, but that way you get a good range of how much bacteria were in that sample. So all of these techniques that I'm going to mention, they all have their strengths and their weaknesses, but we'll go through some of them. So spread plating, optical density, hemocytometer, fluorescence microscopy, flow cytometry, these are all different techniques for counting bacteria. We talked about spread plating already, so you know, if you have questions, you're welcome to, I can go over it. But for optical density, this basically measures growth in real time. So you have bacteria in a tube of liquid, so you can only use optical density using a liquid culture. So you have a tube of liquid. And basically what you do is you put, you know, some of the bacteria in a tube of liquid, and then you put it in something that we call a spectrophotometer, this machine that basically has a light source, and the light source hits through the tube, and depending on how cloudy this tube is, it's able to do a calculation, and then it basically, the cloudier the tube is, the more there are, the more cells there are in that liquid culture. So obviously the cloudier, the more cells there are, but this uses scatter and the light scatter in order to give you a number to tell you an approximation of the cell density. One of the problems with this is that when you are using optical density to count the number of bacteria in a tube, there are also cells that are in there, but they're dead. And so it also accounts for dead cells. So those are also contributing to how cloudy or turbid it is. And so that's going to alter, the optical density. So it's good, but it has its cons as well. So here we have a graph over time where basically we're measuring how much bacteria is in this tube and how fast they're growing over time. So at time 0, there's no bacteria. At 2 hours, you go back and you put the same tube into the machine, and it sees that, okay, there's more cloudiness growing, so the optical density goes up, and then you see, at another time point, it goes up, and so it keeps on growing over time. And so then it kind of stabilizes at the end, because eventually they run out of nutrients in the tube, and so they've grown as much as they can. And so so this is a general curve of bacterial growth, and it's measured by OD, optical density. 600 has to do with the wavelength that's used, but you guys don't need to know the details of that. But does that make sense? Optical density has to be a liquid culture. Basically, light is being measured based on how it scatters and how cloudy and turbid the liquid culture is. And then the con is that you essentially are going to it accounts for dead cells as well. It's quick, it's easy, so it's used a lot. So kind of to just counter this, growing cells on a plate obviously takes more time because you need to give the cells time to grow on the plate, but on a plate you're not going to have dead cells growing. So that's one of the advantages of counting using plating. Any questions on this? Okay. So a hemocytometer, basically you're able to directly count the number of cells. So you have a glass slide, and it actually has this little grid on it that's it's microscopic. You can only see it under the microscope. And so this grid system, basically you put a small amount, a known volume of your liquid, usually it's 10 microliters, it's very, very small. And then you look under the microscope, and usually you will put a dye in there, and the dye will attach to living cells and not dead cells. And so you're able to count the number of living cells, and you count them basically in a grid system. So instead of counting the entire thing, you can count like a few of them and then multiply by how many of these grids there are to get an approximate estimate. So sometimes, you don't put a dye in there, and then you can see both dead and living cells. So it could, depending on what you're using and if you're able to stain it, this one could also not account for just living cells. It could also account for dead cells unless you put a specific dye in there. But by doing that, and because you know the volume that you're putting in here, you can count backwards and calculate the initial concentration in your sample. So if I have a tube that's 1 milliliter, and I take just 10 microliters, so that's very, very small amount, that's 1/100th of a milliliter, and you put that in there, and then you count how many cells are in 1/100th of the 1 mil, then you can work backwards and multiply by 100, and you get the initial concentration in that 1 mil tube. Does that make sense? Okay, so yeah, this is what it would look like, and so there's like multiple of these when you look under the microscope, and so you can like pick a couple of them and count how many are in here, and then multiply by the number of these that you see, and then you can work backwards to calculate the initial concentration. next one is fluorescence microscopy, where you're directly counting live and dead cells. Basically, you distinguish dead cells from living cells by adding a dye. Usually it's a chemical dye. And so here, just an example, all of the live cells are get this dye. This dye enters just live cells, cyto9, and then propidium dye only enters dead cells, and so you can then be able to distinguish between the two. So they fluoresce. Okay. So the previous techniques that I showed, they require you to isolate the sample and then count them and then back calculate the initial, to find the initial concentration of the cells that you are working with. But flow cytometry is different. You're directly counting your cell sample without using microscopy, and you're using an electronic technique that basically will count and separate the populations of your cells according to certain properties, distinguishing properties. So like fluorescence intensity and cell size. So basically you put all of your cells in this sample stream, you put them in here, and it goes through, this machine, and then in this machine it sorts them, basically, and it collects them into these tubes, so based on size and based on their ability to fluoresce. So those are the two things that flow cytometry takes into account. It also sorts cells that it knows are waste, and so it just takes those into another tube. So there's these cell collectors. So cells can be sorted by fluorescence, like if they're live, if they're dead. And so if they're dead, it'll probably take them into the waste bucket. And then side scatter basically correlates to cell size. So usually the way we look at this type of graph, this one is looking at fluorescence, and this one's looking at cell size. So there are larger cells over here because they fall on the right side of the scale, and these are the smaller size cells. And because you're looking at fluorescence level on the y-axis, the lower you are, they're non-fluorescent. The higher you are, they are going to be fluorescent. So does this make sense? Are you guys able to read this kind of graph? So if I were to ask you, if a cell is over here, if I were to circle one of these and ask, what's the size of this and does it fluoresce? You should be able to say, well, it's large because increasing cell size is this direction, and fluorescence, the higher you are, the more it's going to fluoresce. Make sense? Okay, so here's just a little video that I think is cool. It kind of shows you this flow cytometry and cell sorting in action. You can see the red and the green depending on whether they're alive or dead. All right, so What is the fastest way to measure cell density? Yeah.
Optical density.
Yes. Can you tell me why?
Because it uses light to measure like the amount of bacteria in a sample.
Yep, exactly. It's really quick. You remember the graph over time. You can do it every couple hours. You just literally put your tube in an incubator. You take it out when you need it. You put it in the spectrophotometer. It uses light. It measures it, gives you a number. You put it back to incubate it for another couple hours. You take it out. It's very quick and easy. Hemocytometer counting is, a little bit, it requires the math. It doesn't just spit out the number for you. You need to back calculate. Streak plating, you need to also back calculate, but you also have to wait for the cells to grow on the plate, so that's going to take time. And flow cytometry is really, really expensive, and it's going to take a lot of time for you to submit your samples and get the information back. So optical density is the fastest way to measure. Does that make sense? Great. So now let's talk a little bit about some less boring things. Bacteria and how they can behave, especially in groups. So quorum sensing is one of the coolest topics, I think, in bacteriology. Basically, it's when a group of bacteria can collectively respond to a change in the environment. So basically, a group of bacteria can recognize the density or the amount of cells that they are with, so that then there can be a collective response to some sort of environmental change or stressors. It's really, really cool. There is a really cool TED Talk by Dr. Bonnie Bassler, and she studies quorum sensing in bacteria. And basically there were these bioluminescent bacteria that hide out in a squid in the ocean, and when you have enough of those bacteria kind of lighting up, they help the squid hide out in the ocean, and then when it needs to fluoresce and move and do its thing, you have the bacteria, once they all start to express the gene to fluoresce, once it gets to a certain number of cells that are collectively lit up, then it allows the neighbors to be like, oh, they're lighting up, let's light up too. And so it's a collective response to some sort of environmental change. So if you're interested in that TED Talk, it's really, really cool. I could upload it later, but yeah, let me write that down. But we'll talk more about this, about quorum sensing in the future, but I just wanted to kind of give you a little preview here. Swarming is another group behavior of bacteria where they can kind of swarm and move across a surface. They move together in groups across a surface. And then biofilms. Biofilms are basically when a group of bacteria adhere to each other and to a surface. So they do this in order to stay near or around a source of nutrients. This happens a lot when nutrients are depleted in an environment, so they kind of just swarm around each other. And I guess swarm is a bad word to use because swarming is different, but they, stick together, and they're able to, and then they all stick to a surface. So swarming has to do with movement. Biofilms have to do with adherence. Does that make sense? Okay, so these biofilms are actually these specialized structures that they form together, and they, they're surface attached communities, they're called biofilms, and so they are constructed by one species, or it can be multiple species of bacteria, and they can, form on really any surface. It can be organic or inorganic, and so sometimes, when there's a lot of nutrients, they can form, and then when nutrients become scarce, they can detach and forage for other nutrients, other sources. There's an array of patterns that can form. Basically, what they do is they're increasing the colony's surface area It allows, for greater access to nutrients, like oxygen, for the millions of cells making up the biofilm, because there's millions of cells here. And so there's got to be something that's allowing them to stick together and to stick to the surface. So what is this extracellular matrix that's holding this biofilm together? Basically, what are the bricks and mortar for biofilms? And so this is really important. E. coli can form biofilms, but there's this extracellular matrix, an ECM, that is made up of two components, Curli fibers and cellulose. And so together, the Curli fibers, these are just protein fibers. They are resistant to things that will denature them. And cellulose is polymers of glucose. And these are also resistant to denaturants, and they're also highly wettable. So it's okay if you wet them, they won't break apart. And together, Curli fibers and cellulose form these very tough extracellular matrices that make it resistant to things in the environment that can break down the bacterial biofilm. So they've got to be held together by something, and these are the two components. So together, they make these extracellular matrices. This is actually interesting because there is a professor at Michigan, Matt Chapman. He studied this, and he basically studies the Curli fibers. And the Curli fibers are made by gut bacteria. This is just a fun fact. You don't have to know this. But they form these amyloids, basically, that our gut makes to then allow for seeding. I don't know if you're familiar with neurodegenerative disorders. Amyloids are prevalent in neurodegenerative diseases, like Parkinson's disease and things like that. So usually when those connections are, patients are, going through some sort of neurodegenerative disease, those amyloids are starting to deteriorate in things. And so there's actually a gut-brain connection there between the Curli fibers that are made in the gut by bacteria and this link to the gut-brain axis where that helps prevent neurodegenerative diseases. So this is being still really widely studied at Michigan and other places. Just another fun fact. Okay. Questions? All right, so biofilms can serve several functions. In nature, they can take different forms and serve different functions for different species. They can hunker down when nutrients are plentiful. They can be used to find shelter from harsh environments like antibiotic treatment. They can be, kind of all gather together to ward off predators like phagocytes. Those are immune cells that try to engulf bacteria. And so what triggers biofilm formation? There's different environmental signals for even in different species, like pH, iron concentration, temperature, oxygen, presence of nutrients and amino acids. So it's important to know the different things that can cue biofilms and some of the different forms that they can take and why they do this. All right, and so there are distinct stages in biofilm development that I want you guys to know. So first, you have the attachment to a monolayer by flagella in this case. The flagella are the little squiggly tails here. They help with movement, and so they attach, and this is initiation. Then you start to have, colonies coming in, and so this is called a microcolony. They're starting to mature. And then when they mature and they kind of start to form these colonies, they start to produce this exopolysaccharide, or EPS. So this starts to form this like glue that allows them to stick together. they mature, and then they, as they mature, they're maintained. They start to communicate with each other, maybe by quorum sensing. And then, once they recognize that they've reached a certain cellular density, then it triggers this binding to each other with this EPS production, and then you end up having this, big biofilm tower. You can have nutrients flowing in between them, but then as it matures and once the nutrients become scarce again, then certain individuals can start to detach and go away and start foraging for some new resources and nutrients. So this is the dissolution step. Any questions on that? Yeah.
Can you have like multiple different kinds of bacteria form a biofilm?
Yes. Yeah. It could be just one species or it can be multiple. Yeah. Good question. Any other questions? All right, well, if there's nothing else, you guys are free to go a little bit early today. Remember, worksheet 2 is due on Friday. Bring it to class.